In this video, we are going to be describing the steps of DNA replication. And remember from the last chapter, we talked about that before a cell can divide, it has to make a copy of its DNA. If you think about a cell splitting in half, in order for the cell, both new cells, to have a copy of the DNA, it has to be copied first so that when the cell divides, a copy can go to each new cell. Let's remind ourselves about the structure of DNA before we start talking about DNA replication. Remember that DNA exists as a double helix. There is a sugar phosphate backbone in DNA. The sugar is deoxyribose. The phosphate groups then form the remainder of the backbone, and we have our nucleotide bases: adenine, thymine, guanine, and cytosine, which are found in DNA. Remember, RNA contains uracil in the place of thymine. And the double helix shape, for DNA to replicate itself, it will have to unwind. We call the strands complementary nucleotides because remember, adenine always bonds to thymine, cytosine always bonds to guanine. So in order for DNA to be replicated, the first thing that it has to do is to unwind. And enzymes called helicases actually unwind the DNA. There are then single-strand binding proteins that hold those strands apart so that they don't rejoin at their complementary pairs and resume their helix shape. We then have RNA primers that match up with the DNA. So an enzyme called primase ends in ASE, which indicates that it is an enzyme. It adds a short strand of RNA to each template strand. And notice that there is a 5', 3' end. One of the strands, when the DNA has unwound, one of them will have a 3' end, the other has a 5' end. So those DNA segments will basically create sort of a starting point for the DNA replication to be occurring. DNA polymerase is the enzyme that makes the new DNA strands. So it starts again at those RNA primers. Those were the starting points. So that little RNA primer, it is a 3' end because DNA can only copy 3' to 5'. So that little segment allows for a 3' start point for that DNA. And the RNA primers then, they're going to provide those 3' ends. So the nucleotides are going to be added. And remember in the last chapter, we talked a little bit about, we saw a video that talked about Okazaki fragments. Those Okazaki fragments are small bits of DNA so that on one strand, which is called the leading strand, DNA synthesis is going to be continuous. So once that DNA starts copying, because it's 3' to 5', it can just continue. But because the other one was 5' to 3', we keep having to have those little fragments put in there that provide those 3' starting points for the DNA replication. And that strand is called the lagging strand. The lagging strand is the one that is 5' to 3', the original strand, and uses fragments to copy DNA in different little segments. Whereas the leading strand, again, is the one that was the original strand was 3' to 5', and so the DNA replication can be continuous. So once we have DNA replicated, there are going to be on the lagging strand some gaps. DNA ligases fill in those gaps. They provide covalent bonding sites between DNA segments so that the sugar phosphate backbone is sealed. DNA replication actually happens at many different points of origin. So when DNA is copying itself, it's not just copying in a single location, it's copying in lots of different places at once. And the reason that that's really important is that we have billions of genes in our genome. So you can imagine how long it would take to copy the entire DNA if it was done just continuously from start to finish. So having DNA copied, starting along at one point, starting along another point, starting along another point, allows for DNA to be copied multiple places at multiple times. Those spots are called origins of replication. So that is where the replication is actually occurring. Here's a clicker question for you. The image at the right shows a cell's DNA before replication. Which of the following best represents the newly created DNA strands after replication is complete? You might want to pause this and think about that. You should have chosen B, because remember that when DNA copies itself, it divides into the two original strands unwind and separate. And so those are the starting points for creating the new strands. So you have an old strand, which would be represented by the black lines, and the newly copied strand is replicated by, the red strand is the new strand, basically, that has made a copy. So the black strands are the old strand. We had two. We separate them, and each one creates a new copy represented by the red strand. So why must DNA replicate before each cell division begins? Remember at the beginning of this video, we talked about the fact that when a cell divides, it actually is separating into two. And so as it moves apart, if that DNA had not been replicated, only one cell would have a copy of the DNA. So the other cell would basically be completely unable to do anything. Oh, sneeze coming. So, excuse me. So in this video, we described the steps of DNA replication.